So with 1523A is, I think, kind of interesting. I like these types of questions. There's a little math involved, but I don't think it'll be too difficult for us. So let's take a look at the problem. So here we are looking at a culture of bacteria. So what bacteria doesn't even matter. It doesn't even matter what bacterium we're looking at. We just have a bacterium, okay, probably E. coli or something like that. And normally, you know, we can take this bacterium and we can spread, you know, if we spread 100 colonies out on a petri dish. Let's say this is a 100 millimeter petri dish, a standard petri dish, and we're looking down onto the petri dish, which is on a table, and this is the table down here or whatever, and we spread it out. We spread out the 100 colonies, and the next day, you know, we should see those 100. Well, we spread out the 100 cells, and each one of those cells lands in a certain spot, you know, on the randomly lands on the plate somewhere. Well, we spread it with a spreader, which, you know, kind of looks like this. And we spread around the cells in a solution of water or something like that. And if we spread it well, they should land randomly along the plate. And then we incubate them overnight, and the next day we can see a little colony. You know, and if you were to look at this colony from the side, you know, it would just look like a little bump, you know, full of just millions of cells. So this is one colony that all came from one original cell. Okay, so now we're working with a bacterium here. So bacterium, and normally this bacterium or the bacteria will grow on minimal medium. So just minimal medium. This bacteria or an individual bacterium, an individual cell will grow on this medium and form a colony. But we have a mutant. And so now this mutant is called a Lu mutant, meaning that we have to add leucine. So let's say we had 100 colonies of this Lu mutant. I keep saying colonies, sorry. We had 100 cells and we spread them out on a plate of minimal medium. Well, nothing will grow. But if we had minimal medium and we added leucine to that medium, this is our plate now. Remember, these are our petri dishes. It's a petri dish here with the media in there, and it's solid because we add a thing called agar to the media, which kind of makes it like Jello. So it's semi-solid, and a lot more solid than Jello. That's because we have to spread the bacteria on it. But okay, so petri dish, agar, petri dish, we add agar here, so it's solid, semi-solid medium. And this one has leucine in it, and okay, we spread 100 of our mutant cells up here, we spread 100 down here, and we let them grow overnight. And okay, so they only grow when we add leucine, and we can count them up, and we have about 100. Okay, so now the next step for this question. So for the next step, we need to talk a little bit about making serial dilutions or just dilutions of bacterial cultures. So how can I start this? Okay, so let's say we have a 1 mil culture of this bacteria here. A 1 mil culture of bacteria. So okay, this is let's go with 10 milliliters. A 10 milliliter culture of the bacterium. And in here, this culture has 1 million cells. 1 million cells per milliliter. Okay, and this is the Lu minus strain, so the medium has the culture had leucine in there, and it has 1 million cells per milliliter, and we have exactly 10 milliliters of the culture. So if we were to take this and spread 1 milliliter onto a petri dish with minimal medium plus leucine, so how many cells would we expect to see? So we're going to spread 1 milliliter from this culture. How many cells will we expect to see? So we should see 1 million. So we're going to spread 1 million cells here. And okay, well, we should see 1 million colonies. After growing, letting the petri dish incubate overnight, the individual cells will form little colonies, and the next day we'll be able to see those with the naked eye. The problem is 1 million cells, that's too many. That's too many. There's not enough room on this plate to hold all cells. They're going to be overlapping. So it's just going to form this one giant confluent colony. Confluent meaning they're all touching, they're all overlapping. So this whole plate is just going to be full of bacteria. So if we wanted to actually, count to make sure we had 1 million cells per milliliter here in this original culture, we would need to dilute it. So the way we normally do that is to make a carefully, organized dilution series. And we can do that by adding 9 mils of water, or even the medium without the culture medium without bacteria. So 9 mils of medium here in another test tube. We take 1 milliliter from here. We add that to there. And so now we end up with 10 mils, right? So 1 milliliter of this culture, and how many cells are in 1 milliliter? 1 million. So we added 1 million here. We added 1 million, transferred 1 million from here to over here. So now that raises the volume to 10 milliliters. So we have 1 million per 10 mil. So that works out to be 100,000 cells per milliliter. So do you see what we did here? We made a 1/10 times dilution of the original culture by taking 1 milliliter of it and moving it over to 9 milliliters of culture without bacteria in it. We diluted the number of bacteria 10 times, or 1/10. So now we have 10-fold less bacteria per milliliter. So if we were to plate 1 milliliter of this culture, of this dilution, we would see how many cells? Well, we wouldn't see the cells, they're too small, but if we incubated the plate overnight, we would see 100,000 colonies. I should say colonies over here. And again, that's way too many. That's way too many. You probably can only count a couple hundred. Maybe you'd be able to make out a couple thousand colonies on a plate, but you probably wouldn't be able to count those. So a couple hundred or a couple dozen is ideal. So we're going to need to dilute this again. Okay, so we will repeat the process. We won't take the 1 milliliter from this original culture here. We'll take 1 milliliter from here. So 1 milliliter from the 1/10, 1/10 dilution to 9 mil of culture medium without bacteria or just water. That'll make a new 10 mil culture. So let me see. 9 mil plus 1 mil of the 1/10 culture. 9 mil of just go with the water. Of the 1/10 dilution, we'll call it. Okay, so what do? We transferred if we took 1 mil, we know we have 100,000 cells in a milliliter, so we transferred 100,000 here to a final volume of 10 milliliters. So that is going to reduce the number of cells by a factor of 10 again. So we'll have 10,000 cells per milliliter. Okay, great. And we can plate that again and again. Okay, what are we going to see? You probably know what I'm going to say here. So we plate 1 milliliter of this. Well, we should see 10,000 colonies forming on this plate. And we can keep doing that over and over and over again until we get to no colonies. We can get to a dilution that's so low that there's actually no bacteria in a milliliter of our dilution. So, okay, that's how the dilution series works, and we need to understand that to be able to understand this question. Okay, so let's take a look at the question. Okay, so with the question now, we are looking at 7 different dilutions here. So let me map that out for us. So we have our culture. So bacterial culture. And this culture had, we added a mutagen, like EMS. Let's say we added EMS. We talked about EMS, a chemical mutagen, in class. And we let it replicate 1 time. So we should see a bunch of mutations now. And this is a culture of our Lu minus mutant. Okay, so we have our original culture here, which we're going to call, what are we going to call this? Let's just call this 1 times. So that's our undiluted culture. And this is our 1/10 dilution. This is another dilution, which in each time we're going down by a factor of 10. So 1 over 1,000. These are the little test tubes. I'm just diagramming the test tubes for us because we're making our dilutions in test tubes. And here we're at 1 over 100,000. And here we're going to be at a 1,000,000-fold dilution. And then one more 10,000,000-fold dilution. Okay, and I can, let's say these all have 10 milliliters in them, except for this one, because this one we removed 1 milliliter. Actually, all of them, right? We always removed 1 milliliter. So all of these actually, it's a complicating detail, but they actually only have 9 milliliters left in them, except for this one, which has 10 milliliters. If you don't understand that, you can go back and look at the, this part up here, where we talked about this part. Okay. Anyways, that was, we made the dilutions, and the total amount of liquid in here isn't that important at this point. So now what we're doing here is we are going to plate some of the culture from some of these dilutions onto minimal medium, a petri dish with semi-solid minimal medium, and some petri dishes with semi-solid minimal medium plus leucine. And so we are going to use this one right here, and we're going to plate this one on minimal medium. So how many should grow? Right? So this is a bacterium, a strain of bacteria that is mutated in a gene required to produce leucine. So none of them should grow on minimal medium, right? But so what we're thinking here is, well maybe, maybe this is like a point mutation, which maybe this gene required to make leucine has like 1 point mutation, just one base was mutated to a or a T or something, and or whatever. And if that mutation reverted back to the wild type copy, mutated again, then okay, all of a sudden this bacterial strain would be able to produce leucine on its own. And that's called reversion. So you can have a gene and it can experience a mutation and it can no longer function, and then it can mutate again. That same base, if it's just one base we're talking about, that base can revert, it can mutate again and go back to the wild type sequence and function again. And here there are millions and millions and millions of cells, and we expose these cells to a chemical mutagen. And what we're doing now is we're looking to see how many of those cells that were mutated in this leucine gene had that gene revert back to the wild type. And let's see. So the problem says, I can copy and paste this problem here. One second. I think I can. Let's see. I'll pause this so I can copy and paste it. Okay, so let's see if this works. Okay, we got it. Okay, here's the table from the problem. And so the problem is telling us with the, okay, this is actually the 1/10. The symbolism is a little strange here. I don't know if this means we're using the 1 times dilution or we're using the, well, let's just say this is the 1 times. We're going to call this the 1 times, the undiluted right here, 10 to the -1. Okay, so when we plate when we plate this out over here, I don't know, we plate like a milliliter, let's say. Does the question say how much we're plating? Let's see. No, okay, let's just say we plated a milliliter here on the plate. It's kind of a lot for a 100 millimeter petri dish, but let's just say we did that. And we see 18 colonies. Okay, well, we weren't supposed to see any, right? 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. We weren't supposed to see any of these, but now we're seeing 18 colonies. So it looks like adding EMS, it's possible adding EMS caused reversion of the Lu- gene, the mutant gene, to Lu+. So it now functions. However, there were millions of cells in here. Most of them did not revert. So most of them are still Lu- mutants. And so we want to determine what is the mutation rate. You know what I mean? How many of those colonies underwent reversion to wild type? So we can measure that by if we knew how many colonies were there in that 1 times dilution. Okay, so how can we know how many colonies were in there? Well, we could simply just plate this out, right? Minimal medium plus leucine. Okay, now everything can grow, right? So we're not going to plate these guys out. We're going to go back to the original culture, right? Plate them at the same time. But okay, there's way too many. There's going to be millions here. Okay, we can't. So that's not very much use to us. But what we can do is plate these guys out down here, and we are going to look at this 10 to the -7 dilution here. Okay, and we're going to plate that out on minimal, medium plus leucine. So we've diluted the number of bacteria in there to a point where we can actually count the colonies. And how many are we counting? 1, 2, 3, 4, 5, 6. So there are 6 here. Okay, so now we just need to do a calculation to allow us to determine how many cells, were in the original culture. So these are cells that are Lu-, and probably none of them are Lu+, because so few are Lu+, but both Lu- and Lu+ cells will grow on this medium. So if we were to plate this one out instead, how many colonies will we expect to see? Well, there should be tenfold more bacteria in this culture over here than over here. So we would see 60. I'm not going to diagram all of these colonies, but we would see 60. The same thing for this one over here. We should see tenfold more. So we should see 600. Over here, we should see 6,000. We'll keep going up. From this one, we should see 60,000. And you can see why we have to plate from this lower number down here, because we can't count this many colonies. We can't do it. But so now I'm just kind of going through here and showing you how many, if we could count them, we would see. So 60,000 there, 600,000 here, and 6 million here. So, okay. So, and if we played it from this one times, we would see 60 million. But now I'm thinking originally I said we played it from this guy, but let's go with 10 to the negative 1. Let's call this 10 to the negative 1 right here. Okay, so to summarize, what are we seeing? We're going to go back, and let me just summarize. So with the 10 to the negative 1 dilution, when we plate this on minimal medium without leucine, we see 18 colonies. 18 colonies on minimal medium, no leucine. Okay, and we went up there. I walked you through what would happen if we had been able to count all of the colonies when they were plated, when the same number or same amount of the culture was plated on minimal medium plus leucine. Okay, let's see, what was that number? 6 million. So 6 million colonies would have been on the minimal medium plus leucine. So we actually had to plate a dilution of that culture in order to be able to figure out, that okay, there are 6 million colonies in that culture. So what does this mean? So let's take a look at the 10 to the negative 1 dilution. So in there are 18 colonies that are Lu plus. There are 6 million colonies total. I'm saying colonies, but cells. 6 million in total that are either Lu plus or Lu minus. So we want to figure out the mutation rate. So because if we didn't add EMS, we would expect that we'd have no Lu plus colonies. So EMS caused mutations at that Lu1 gene, at that specific gene, at a certain rate. And what is that rate? Well, we're just going to take that 18 and we're going to divide that 18 mutants, or revertants in this case. 18, sorry, having trouble spelling as usual. Revertants divided by 6 million cells is going to give us a certain rate, and I'll take out my calculator here, see if I can find it a little faster than I found it yesterday. You think it would pop up right away because I just used it yesterday. Okay, so 18 divided by 6 million. I think there's going to be 3 in there somewhere. 1, 2, 3, 1, 2, 3. That's 6 million? Yep. There isn't a 3. Okay. Anyways, I stand corrected. Okay, 18 revertants divided by 6 million cells is going to Is that right? Yes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 is going to equal 5 times 10 to the negative 13. That doesn't seem right. That doesn't seem right at all, does it? Did I do something wrong? I think we needed to do it the other. Let me see. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 5 times 10 to the negative 13th revertants per 6 million cells. That does not seem right. Okay, hold on here. Okay, so I guess sometimes it's fun to watch your professor struggle with these problems, right? And okay, if this was right, this would have been per million cells. But it's wrong, and I look back at the video and something happened with the calculator. I don't know what was going on. Maybe you caught it. But let's take a look again here. I'm going to get the calculator. And okay, so 18 divided by 6 million. So 18 million revertants divided by 6 million cells equals, okay, there we go. I knew there should be a 3 in there. So 1, 2, 3, 4, 5, 6. Okay, let me count that again. 1, 2, 3, 4, 5, 6. Okay, so we calculate that correctly. We should have 3 times 10 to the negative 6 revertants, and we can consider that mutants too. We can just say 3 times 10 to the negative 6 mutations per cell. Okay, so what does that mean? So essentially it means we have to have a whole bunch of cells. If we have a whole bunch of cells, a certain number of them are going to have a mutation at that locus. So we can actually say, let's say we have 18, let's say we have 6 million cells, right? 6 million cells, and we can multiply that by our mutation rate. Our mutation rate is now 3 times 10 to the negative 6 mutations per cell. And let's see how many we get here. So 6 million cells times, let's see, I don't know if we have an exponent here. We do. Exponent negative 6. Yes, okay, we got it. Okay, so we can take a known number of cells and we can figure out how many of them should have a mutation in that Lu gene that reverts it back to wild type, because we figured out the mutation rate of 3 times 10 to the negative 6 mutations per cell. Essentially it means we need a lot of cells to pick up a mutation there. So I also just took a look at the answer in the solutions manual, and the author of the solutions manual wants us to divide this by 2 because the cells replicated after they were treated with the EMS. And I say that's just crazy. We're not gonna do that. We're gonna stick with this number right here. Okay. So okay, we're going to go on to the next problem now. The next problem is going to be 15.24. Okay.